Okay. So can anyone quickly confirm if the screen is visible? My PowerPoint is visible to you all?
Yes, sir.
Okay. Okay. So, uh, yeah, it's there. So good evening all. Welcome to today's week 4 problem solving session. I'm Rajesh. I'm a PMRF research scholar at the Department of Biotechnology, IIT Madras. And in this session, we'll mostly talk about the cell organelles, and the cell division like mitosis and meiosis. Most of the things you have, I think, you have already studied in class 11th and 12th, that label. So that's kind of brushing up your old concepts. And instead of like giving a what do you say, big definitions for mitosis and meiosis, I'll just simply draw a diagram, and that will make things more clear. And so we'll start by discussing what is a cell. So you have came across this basic definition of cell, that it is a structural and functional unit of life. So everything that is living is made up of a cell, either a single cell or a multi-cell, like more than one cell coming together and performing a single function. So, since cell is considered the basic unit of life, because they come in discrete and easily recognizable packages. So, it comes like this type of packages, because the cells are surrounded by a structure called a cell membrane. So, on the left, you can see it is a more advanced cell, which is a eukaryotic cell. On the right, you can see a prokaryotic cell, which is a primitive cell. But the to be a cell, there has to be a cell membrane. So if there is cell membrane, so that's like the clear boundary between the cell's internal and the external environment. Okay, so that is the definition of a cell. So as we saw, there's two type of cell. One is prokaryotic cell, one is eukaryotic cell. In the course of evolution, also we talked that first these prokaryotic cell evolved, and from them these eukaryotic cells came. So since these are prokaryotic or very primitive cell, these has to be not so advanced. These has to be very basic in structure and in function, and these has to be unicellular organisms because they are primitive organisms. So, who are the members of prokaryotic? We read this in week 2 during our discussion on classification, that it has bacteria, blue-green algae, and mycoplasma. And all have a cell wall surrounding the cell membrane except in mycoplasma. Mycoplasma do not have any cell wall. Other than the mycoplasma, everyone has a cell wall. And the characteristic that defines prokaryotic cell is that genetic material is basically naked, not enveloped by a nuclear membrane. So the genetic material is not enclosed in a nuclear membrane. In addition to the genomic DNA, sometimes their single circular DNA is also found in many bacteria, and that is called as plasmid. So if this is the main genomic DNA, some extra chromosomal circular DNA is found, that is called as plasmid. Have you people came across plasmid? That plasmid is used in genetic engineering. We take out plasmid, and there we put our gene of interest, and then we give it back to the bacteria, and the bacteria can multiply the plasmid. So plasmid is a extra chromosomal self-replicating structure. Remember this. This comes in MCQ many times. Plasmid. I I don't know if there's more discussion in the subsequent things about plasmid, but I hope you know about this plasmid and the isolation of plasmid. And the ribosome, in the prokaryotic ribosome, we all know that is it is a 70S ribosome. So S stands for Swedberg unit, the sedimentation coefficient. So the larger subunit is 50S and the smaller subunit is the 30S. And in total, they make a 70S. So you don't tell that 50 plus 30 is not 80, then why you are writing 70? So it is not a like subtraction and addition, it is depends on the sedimentation coefficient, how long it is taking to sediment or settle down. So total it takes 70 years. Inclusion, it has inclusion bodies that stores the reserve material in the cell. So inclusion body are not bound by any membrane and lie free in the cytoplasm. That is phosphate granule, glycogen granule, and they have mesosome. So mesosome is another characteristic of the prokaryotic cell. So mesosome is nothing but like the extension of the plasma membrane into the cell. So if this is the continuous plasma membrane and we have some extension like this, then this is called mesosome, and it kind of forms vesicle and helps in the cell division thing, those kind of function this forms, and these are called as mesosome. So the main thing is they have a cell wall, and since bacteria consist most of the prokaryotic cell, and their cell wall, we have to know about their cell wall. So their cell wall is made up of comes in MCQ, peptidoglycan. So how you distinguish bacterial cell wall? It has peptidoglycan. Peptidoglycan, polysaccharide, and teichoic acid. In many MCQ, they will ask what is there in the cell membrane. They already gave peptidoglycan, polysaccharide, and this is the option, teichoic acid. So remember, peptidoglycan, teichoic acid, and protein polysaccharide are the cell wall components of a bacteria. And the peptidoglycan is a monomer of, I'll show you the structure later. So it is a monomer of N-acetylglucosamine and N-acetylmuramic acid. So it is a continuous chain of NAG and NAM. What is this peptidoglycan? So this is a very basic overview about prokaryotic cell. Now I'll move to the next advanced type of cell, that is eukaryotic cell. So in eukaryotic cell, because they advanced like in the course of evolution, they came later and they it has to be more efficient, or like let's say more complicated than the prokaryotic cell. So on the left, you will see the animal cell. On the right, you'll see a plant cell. So plant cell, you all know that they have a cell wall, not the animal cell. Animal cell don't have a cell wall. So who are the members of eukaryotes? So we have protist, plant, and animal and fungi. These are the member of the eukaryotic cell. I mean, these kingdom have eukaryotic cell. So eukaryotic cell, there is an extensive compartmentalization of cytoplasm through the presence of membrane-bound organelle. So this is the characteristic feature of eukaryotic cell. You get membrane-bound organelle. Plant and animal cell, as I said, they differ that the plant cell has a cell wall, plastid, and a large central vacuole. Plastid we'll discuss later. And characteristic of plant cell is that they have a large central vacuole, and this vacuole can sometimes take up to 90% of the cell, whereas animal cell either do not have a vacuole or if they have very small vacuole, depends. So that is the characteristic of the plant cell. They have a cell wall. And what is the characteristic of animal cell? Like what sets them apart is that the centriole. Animal cell have centriole which helps in the cell division. We'll see how they help in the cell division, which are absent in plant cell. So these are the two contrasting features between animal cell and plant cell. Plant cell has uh, cell wall, a large vacuole, whereas animal cell has centriole. We will now subsequently look into the major organelles of the cell, starting with the, oh, sorry, before that. In the previous slide, I discussed about the cell wall of the bacteria. Since plant have a cell wall, we'll have a look at the plant cell wall. So there I said it is made up of peptidoglycan, polysaccharide, teichoic acid, and the proteins, of course. But in the plant cell wall, it consists of cellulose, hemicellulose, pectin, and protein. So which sets them or makes them different is that cellulose, hemicellulose, and pectin. So you can see plant cell wall in this picture. So there's a middle lamella. There's a middle lamella which is represented here in the white cement-like deposition. It is mainly made up of calcium pectate, which holds or glues the different neighboring cells together. So it is kind of a cementing material. Name is middle lamella, made up of calcium pectate. And the cell wall and middle lamella may be traversed by plasmodesmata. There may be some bridge or gap between two cells, and this is called plasmodesmata, and it is used for the transfer of substance material or sometimes genetic material, uh, and that is the role of the function of plasmodesmata. So you can see the plasmodesmata here. Bottom line is the cell wall in the plant is made up of cellulose, hemicellulose, and pectin. So the next I will discuss briefly about the major cell organelles of a cell, starting with the cell membrane. Cell membrane, you all know we have a fluid mosaic model, which is the accepted model for the cell membrane, which was given by Singer and Nicholson in the year 1972. So, according to the name of this model is the quasi-fluid nature of the cell membrane. So, it tells us that the proteins are floating around in the sea of liquid bilayer. Okay. So, the quasi-fluid nature of lipid enables lateral movement of protein within the overall bilayer. One of the most important function of plasma membrane is the transport of molecule across it. So depending on the need of the cell, what the cell needs, uh, it forms as a barrier, and it form the protein embedded in the plasma membrane functions the role or act as transporter. Either there can be passive diffusion or passive transport without the use of energy, or there can be active transport with the use of energy. So, what is a lipid bilayer? So you can see these are the lipid structure. The name of the lipid is not just simple lipid. It's phospholipid. So it's phosphate. It has a phosphate head and a fatty acid chain, and it arranges in this type of layer, and not just one layer, it arranges in the two layer, the hydrophobic end facing towards each other. That is why it is called a phospholipid bilayer. So the hydrophobic means the water repelling, or the or not liking the water. This chain, fatty acid chain, is towards the inside, and the hydrophilic or the water liking part is towards the outside. So if this is extracellular, this will come in contact with the environment, and if it is the intracellular, it will come in contact with the cytoplasm. Okay. So this is about the cell membrane, the semi fluidic or the quasi-fluidic plasma membrane given by Singer and Nicholson. Okay. So the second one is composed of four members. Why we and these are studied together. So it is called endomembrane system. These organelles are considered together as an endomembrane system because their function are coordinated. All this function of the four are kind of coordinated, like one comes after another. So that is the reason why we study them together. And the members of this endomembrane system are endoplasmic reticulum, the Golgi complex, the lysosome, and the vacuole. So we have four members in the endomembrane system. Remember this: endoplasmic reticulum, Golgi apparatus, the lysosome, and the vacuole. Just 1 second, I just need to take the attendance. Okay. So a brief function about the endomembrane system. What does this endoplasmic reticulum do? Endoplasmic reticulum has, this small circles, green circle you can see here, are ribosome. And as you know, the ribosome at the site of protein production. Ribosome read the mRNA, and from mRNA they put together the amino acid and make the protein. So there's two type of endoplasmic reticulum, in which one has the ribosome attached and one in which the ribosome is not attached. The ribosome attached is rough endoplasmic reticulum. Ribosome not attached is a smooth endoplasmic reticulum. So in this page, I have put together all the function of these four: endoplasmic reticulum, Golgi, lysosome, and vacuole. As I said about endoplasmic reticulum, we have rough and we have smooth. So rough, as they have ribosome attached, so they are involved in the protein production and some minor folding also. The smooth endoplasmic reticulum is the site of the synthesis of lipid. This is where lipid synthesis happens in the smooth endoplasmic reticulum. So next comes Golgi apparatus, which is in close proximity with the endoplasmic reticulum. So the protein, once it passes through the this endoplasmic reticulum, particularly rough endoplasmic reticulum, migrates towards the Golgi apparatus. The Golgi then perform the function of packaging, and it acts mainly as a packaging material. So to deliver it either to intracellular target or secreted outside the cell. Material to be packaged in the form of vesicle from the endoplasmic reticulum. So once we are done with the things are done with the rough endoplasmic reticulum, it will migrate towards Golgi, towards the cis phase. So Golgi apparatus has two phases. One is cis, one is trans. It will migrate towards the cis phase. The cis phase is towards the endoplasmic reticulum. And how it will migrate from this rough endoplasmic reticulum towards the Golgi apparatus in the form of transport vesicles. In this transport vesicle, it will migrate towards the Golgi apparatus and go and fuse with the cis phase. So this is what is written here. Then the protein are modified in the cistern of the Golgi apparatus. This is the inside of the Golgi apparatus is called cistern. So the protein will be modified and they will be packaged and they will be subsequently uh, be coming out. See, before they and then they will be subsequently released from the trans phase. Sorry, this is the trans phase. From here, they will be released as the secretory vesicles. And Golgi apparatus are also the important site of formation of glycoprotein and glycolipid. So I use this word here, that protein modification. So see, the protein and lipid produced in the endoplasmic reticulum were glycosylated in the Golgi apparatus according to their need, and now they have become glycoprotein and glycolipid, and then they can go to their respective destination in again vesicles, secretory vesicles. The third one is lysosome. So lysosome are membrane-bound vesicular structure that are formed by the process of packaging in the Golgi apparatus. So lysosome are kind of formed in the Golgi apparatus itself, and they are vesicle bound structure. And what there is inside, they are rich in hydrolytic enzymes like lipases, proteases, carbohydrates, hydrases. Since they have lipases, proteases, carbohydrates, that means they can digest lipid, protein, and carbohydrate, and these are optimally active at the acidic pH. See, that is what it is written. that these are capable of digesting carbohydrate, protein, lipid, and nucleic acid. So that is the function of lysosome. The third one is a vacuole. So vacuole, as I said, is mainly found in the plant cell. If in animal cell, these are either not found, if found in, they are very small in size. So the vacuole is membrane bound, space found in the cytoplasm. It contains water, sap, excretory product, some toxins, and other material which are not useful for the cell. And the vacuole is bound by a single membrane called as the tonoplast. The membrane of the vacuole is called tonoplast and can occupy up to 90% of the volume of the cell. So these four form endomembrane system because their function is more or less coordinated. So that's all for the endomembrane system. Here we studied endoplasmic reticulum, Golgi, lysosome, and vacuole. You may be wondering why this vacuole is doing here because these three more or less their function is aligned. But let's say if something is toxic to the cell, like there's too much of lysosome or peroxisome, it or like some waste material or the protein which is not directly used, they can go to the vacuole and stored as some toxic material or sometimes like as a product which is not used by the cell. So that is the reason why they are studied together. It's the endomembrane system. Okay. I hope till here everything is clear. Anyone would like to ask anything? Okay. If not, we will proceed further to mitochondria. So mitochondria, you all know, is the powerhouse of cell. And in an offspring or in a organism, whatever the mitochondria is, comes from the mother. So typically, it is a sausage shape or a cylindrical shape having a diameter of zero point 0.2 to 1 micrometer, average is zero point 0.5, and length of the mitochondria varies from 1 micrometer to 4.1 micrometer. Mitochondria is a double membrane bound structure. Can anyone here just tell what is the other? That's two other, but can anyone tell what is the other double membrane bound structure? Anyone? Which is another double membrane bound structure? Organelle, which is a double membrane bound organelle apart from mitochondria. You can put it in the chat box also. Okay, so just one question to be clear. Are you people reading this organelles? You people have already read about the cell organelle in your plus two, right? In your class 11th, 12th. Have you people read about the cell organelle before?
Yes, sir.
Okay, so I'll quickly tell. So apart from mitochondria, we have nucleus. Nucleus is also double membrane bound, and we have chloroplast. That is also double membrane bound. Okay. So we'll continue with the mitochondria. It is a double membrane bound structure and the inner compartment is filled. So you can see one is outside. Okay. One is this outer membrane and one is inner membrane. And in the inside is filled with something blue color of matrix and that is called as matrix. So mitochondria, how they are or why they are called as the powerhouse of the cell. because it is the site of aerobic respiration. They produce cellular energy in the form of ATP, and that is why they are called powerhouse of the cell. The matrix of the mitochondria possess single circular DNA molecule. And I hope you are clear about this because I asked in the second TA class, that what mitochondria DNA is used for the evolutionary analysis. And that is the mitochondrial DNA because the mitochondrial DNA does not undergo recombination and etc. And that is why they are kind of preserved. So mitochondria has its own DNA, single circular DNA molecule, few RNA molecule, and also ribosome. This is important about the mitochondria. Inside the mitochondria, it has its own circular DNA, few RNA molecule, and ribosome, and these ribosome are 70S ribosome and are the component required for the synthesis of protein. So mitochondria can synthesize its own protein. Uh, we will, of course, we'll have a detailed overview about this, how the mitochondria is called the powerhouse of the cell because it produces ATP. Just a quick overview here. Mitochondria is able to produce or ATP because of this mechanism. F0 F1 particle or the ATP synthase, which is present in the inner mitochondrial membrane and towards facing this F1 facing towards the matrix of the mitochondria. So as I said, there are two membrane or the mitochondria is double membrane mount. One will be here outside and the one is this one, the inner mitochondrial membrane, and the F0 is embedded in the inner mitochondrial membrane and the F1 is facing towards the mitochondrial matrix. And this intermembrane space is where there will be H plus or the proton gradient will be produced. And this proton gradient, how it will be produced? By electron transport chain, ETS. So anyway, we'll be having a discussion on that. That is a completely different section of discussion. So by electron transport chain, we will be having a lot of H plus ion or proton gradient, which will be produced in this intermembrane space. Then this proton gradient will cross this F0 and go towards F1. And in that process, ADP will combine with phosphate to produce ATP, adenosine triphosphate. Diphosphate will combine with the phosphate to produce the adenosine triphosphate. So, as I said, F0 and F1 particle, F0 embedded in the inner mitochondrial membrane, F1 facing towards the this matrix of the mitochondria. And for each ATP produced, 4 H plus or 4 proton passes through the F0 from the intermembrane space to the matrix down. Remember this some many times asked in the question that in the ATP synthase or in the F0 F1 particle in the mitochondria, how many H plus ion pass for one ATP production? For one ATP, 4 H plus ion pass through this. So this is the ATP synthase, which is located in the mitochondria. H plus ion is generated by electron transport chain. H plus ion pass through, we get ATP. ATP is the currency of energy that is used for so many functions, mostly respiration and used for the food, carb, glucose synthesis or glycolysis, you can say. Okay, so that is all about mitochondria. Fourth, we'll be discussing about plastid. So plastid are found in the plant cell and in euglenoids. Why euglenoid? Because chlorophyll or chloroplast is also not chlorophyll. Chlorophyll is the pigment. Chloroplast is the organelle. So is also found in euglenoid. So plastid are found in all plant cell and in euglenoids. So based on the type of pigment, what type of pigment is there, the plastid can be classified into chloroplast, chromoplast, and leucoplast. So chloroplast, as you can like guess, it has chlorophyll and carotenoid pigment, which are responsible for trapping the light energy essential for photosynthesis. Chromoplast, on the other hand, has fat-soluble carotenoid pigments like carotene, xanthophyll, and other are present. Since pigments are present, they give the color to the plant. Yellow, orange, and red based on what type of pigment is present. So chromoplast is for the pigmentation. Leucoplast are colorless plastid of varied shapes and size with a stored nutrient. So there are three types of leucoplast. One is amyloplast, one is elaioplast, and one is alluroplast. Amyloplast store carbohydrate or starch, that is potato. Elaioplast store oil and fat, and alluroplast store protein. Based on the material stored, they are named accordingly, and these are the leucoplast. Okay. So one plastid we need to discuss, that is chloroplast. Chloroplast because they can here you can see they can trap light energy and are essential for photosynthesis. So if you zoom into a chloroplast, so it looks like this. It looks like the stack of coin, and the stack of coin are interconnected among themselves. And the matrix inside this chloroplast is called as stroma. So there's a number of organized flattened membranous sac. They are called the thylakoid. So imagine the stack of coin. So each coin is called thylakoid and are present in the matrix that is stroma. Thylakoid are arranged in stacks like a pile of coin, and this one pile of coin is called as grana, or singular is a granum. In addition, there are flat membrane tubule called as the stroma lamellae. So they are interconnected by this stroma lamellae. So you can see the stroma lamellae here, the connections by the stroma lamellae connecting the thylakoid of the different grana. The membrane of the thylakoid encloses a space called as the lumen. So each coin is called as thylakoid, and whatever is inside a coin is called as the lumen. And the stroma of the chloroplast contains enzyme required for the synthesis of carbohydrate and protein. It also contains small double-stranded DNA molecule and ribosome. So just like your mitochondria, the chloroplast has also its own circular DNA molecule and ribosomes. Very important. So the where exactly the chlorophyll is present. So chlorophyll is present in the coins or in the thylakoid. So in this single coin, whatever you are seeing, in this the green color pigment or chlorophyll is present. So how this uh, this thylakoid so thylakoid makes a stack, or the one stack is called granum. So how this helps in the capturing of light and doing the photosynthesis. Uh, I hope you have seen this type of system before. So these are called the light harvesting complex, LHC, which are present in the thylakoid. So these are the light harvesting complex. And when the light from the or the ray of the sun hit these light like LHC, the electron from this goes to a electron acceptor above the LHC, and then the further the electron is circulated in an electron transport chain. just like a electron transport chain in the mitochondria. And in that electron transport chain, this ATP is produced, or the energy, the currency ATP is produced. And from the electron transport chain, like it goes to the light harvesting complex of photosystem 1, and then again it gets to another electron transport, and then goes to NADP plus, which becomes NADPH. I hope you all know that ATP and NADPH are the currency of energy, or are required for the production of uh, further, or are used later in the process of food production by the plant, ATP and NADPH. Something called as the light reaction and something called as a dark reaction. Whatever I am showing you here, there is light or there is sunlight. So these are happening in the presence of light, hence called as light reaction. And the order on in which the light reaction happens is called as the jet scheme of light reaction, because first the photosystem 2 uh, passes on the electron, then it goes to the photosystem 1. And these are named photosystem 2 and photosystem 1 because, can anyone tell why it is not like photosystem 1 at first and photosystem 2? Okay, this has nothing to do with their name. So if electron goes from here first, then why it is not named as photosystem 1? So thing is, they are named in the order of their discovery. So scientists first discovered photosystem 1, and they named them as photosystem 1, thinking that there is the only one photosystem. But actually, later they found there is not one, there is two photosystem. So they named it later photosystem 2. This might be confusing in MCQ, that which one excites or supplies the electron first in light reaction. It is photosystem 2. And the light harvesting complex of photosystem 2, the optimal wavelength for the electron to jump is 680 nanometer. And for the photosystem 1, it is 700 nanometer. So if it is providing electron, electron is going to the acceptor, then going to the electron transport system, then going again, and then going to NADP. Then you may ask, from where this light harvesting complex is continuously supplying electron, from where it is bringing this electron to give to electron acceptor and further to electron transport system. So the plant absorbs water. So the water is split, and this see this reaction, it is generating electron continuously, it is generating electron. So this electron is supplied to the light harvesting complex of the photosystem 2, and this electron is further excited and goes to electron acceptor and further goes down to electron transport chain. And we get ATP and NADPH in the light reaction. So in the light reaction, this ATP and NADPH is further utilized in the dark reaction. And in on your right side, you can see light dark reaction, where this ATP, dark reaction does not mean it occurs only in the night. It means that it is not depending on the light, sunlight. It is depending on the product of the light reaction. So on your right, you can see we are using this ATP and NADPH and producing sucrose or starch or food for the plant. Can anyone name what is the name of this cycle? Very famous cycle. I'll just zoom. Can anyone just name what is the name of this cycle? If people can try, what is the name of this cycle? Anyone?
TCA cycle or Krebs cycle.
Krebs cycle occurs in the mitochondria. This is Kelvin cycle, right? This is Kelvin cycle, which occurs in the plant. We are talking about plant. That is TCA is tricarboxylic acid cycle. This is your Kelvin cycle. Okay.
Okay, sir.
Okay. I mean, you people have studied this in class 11, 12. These are in the 11, 12 standard. You just have to memorize it. From here only, you will get the questions. Okay. So, the heart of the cell is the next, that is the nucleus. So, why it is the heart of the cell? Because nucleus has all the genetic material and it has this ATGC, and this ATGC is read and made into protein that expresses the phenotype, and it decides what the organism will do, how the organism will look. So all the genetic information is inside the nucleus. So nuclear envelope consists of two parallel membrane with a space between the two membrane 10 to 50 nanometer called the perinuclear space, and it forms a barrier between the material present inside the nucleus. It is what is it is inside the nucleus, and the outside is, of course, outside is cytoplasm. So here, this is cytoplasm and this is nucleoplasm. Cytoplasm of the nucleus is called nucleoplasm. Okay, so it has nuclear pore that are act as passage through which movement of RNA and protein molecule take place. You all know that DNA, by virtue of transcription, makes RNA. RNA comes out of this nuclear pore, goes to the cytoplasm, and in the cytoplasm, RNA is read by the ribosome and protein is made. So site of protein production is cytoplasm. For that, the RNA which is made in the nucleus has to go out. So that this is the central dogma theory that DNA makes RNA, RNA makes protein. So the things that has to be exchanged between the nucleoplasm and cytoplasm, we need pores, and so there are nuclear pores. So as I said, nucleus ha has nucleoplasm, and nucleus has something which has very dense type of nucleoplasm, and these are generally not stained since these are very dense, and this has a special name which is called nucleolus. So the nucleolus is the active site of ribosomal RNA synthesis. So as you know, we have three types of RNA: mRNA, tRNA, and rRNA. rRNA is the ribosomal RNA needed for the synthesis of ribosome. So that is synthesized by the nucleolus. Here, okay, in this above picture, I told you the nucleus has genetic material. So the genetic material is kind of intertangled in a normal cell which is not dividing. So in the picture you have seen this, that this is how the chromosome look with the sister chromatids, but this is not in a normal cell. Normal cell means a normal cell which is just metabolizing. This type of chromosome structure you will only observe in the cell that are going to divide, or the dividing cells. So this is what I want to tell you, that chromatin is something which is uncondensed form, or which is kind of scattered around the nucleus. Then when the cell decides to divide, the chromosome condense and they form this kind of structure called as the chromosome. They have the sister chromatid, each sister chromatid, and the say they have the kinetochore where the centromere come from the centrosome, and then they attach, and then they separate these two sister chromatids. So that is like a basic overview about the nucleus and the nuclear envelope, the nuclear pore, and how the organization of the genetic material in the nucleus. We'll have a look more about have a discussion about this genetic material in context of mitosis and meiosis. Okay. So quickly answer these questions based on these concept studied till now. The first one, the part of eukaryotic nucleus that regulate the exchange of material with cytoplasm, lamina, pore complex, chromatin, or the nucleolus. You can put in the chat box also.
The nuclear pore complex.
Okay. Others, please try to answer these in the chat box also. Very simple questions. Okay, correct. So these are the nuclear pore complex for the nuclear pore, which helps in the exchange of material between the nucleoplasm and the cytoplasm. Option B. Second question. The role that mitochondria plays in the programmed cell death or apoptosis. Can you tell about this, or I'll also tell about this more? Anyone knows the answer to this one? Generate ATP for cellular activity, or synthesizes ribosome for protein production, or produces reactive oxygen species ROS, or release cytochrome C to trigger caspase activation. So which one is the mitochondrial role in apoptosis? Okay. So people can try in the chat box also. It's okay if it is wrong. I mean, okay, fine. I'll tell you this one. So this is option D. It releases cytochrome C to trigger the caspase 9 activation. I hope you all know what is apoptosis. Apoptosis is programmed cell death. Necrosis is that you are making the cell to die. You are like injuring the cell, it is dying. Apoptosis is like the formation of digits or the finger. So when the finger or the is formed, all the fingers will be intertwined, like kind of a net-like structure. So what decides that there will be gap between the finger? That some cell in between the finger has to die so that there will be gap between the finger. That is called as the programmed cell death, and that is called as apoptosis. One role mitochondria plays is that they release the cytochrome C. So I said about the electron transport chain or the ETS that is happening in the inner mitochondrial, sorry, this intermitochondrial space. Mm. And that is how it is enriching H plus ions. And this H plus ion or the proton then cross the ATP synthase and then the ATP is produced. So we will talk about the complex which are there in the electron transport chain later. Just for your overview, it has complex 1, 2, 3, and complex 4. And between this complex 3 and complex 4, there is something called a cytochrome C. So cytochrome C is also an electron acceptor, and it transfers the electron from the complex 3 to complex 4 in the respiratory chain or in the electron transport chain. So in the cell death, when there is programmed cell death, or wherein when there is apoptosis, this cytochrome C is released to the cytoplasm or the cytosol when a cell is activated for an apoptotic stimuli, and this triggers the formation of apoptosome, which activates the caspase 9. So caspase family of protein are a family of protein that are related with the apoptosis. So they make the cells to undergo apoptosis. So this cytochrome C goes and activates the caspase 9 and then leads to the cell death. So this is the mechanism of mitochondria in the context of apoptosis by the use of cytochrome C. Okay. So clear about this? Then we'll go to the next question about chlorophyll. Just now studied about the chlorophyll. So the chlorophyll in the plant cell contribute to the energy production by converting chemical to mechanical energy or storing energy in vacuole or capturing light energy for photosynthesis or facilitating glycolysis.
Then option C.
Option C. Okay.
Option C.
Option C. Correct. So it captures the light energy for the photosynthesis. Correct. And who does it for them? The thylakoid does it for the chlorophyll. Sorry, the chloroplast. It has chloroplast has thylakoid. Thylakoid has chlorophyll. Chlorophyll are the green color pigments which has this light harvesting complex. And by this light harvesting, using this light harvesting complex, they can capture light for photosynthesis. So option C. Okay. The next one. The structure that is absent in the prokaryotic cell, but it is present in the eukaryotic cell. Ribosome, chromosome, membrane-bound organelle, or peptidoglycan layer. What is present in the eukaryotic cell?
Membrane bound organelles, sir.
Membrane-bound. Okay, correct. So membrane-bound organelle is a characteristic feature of eukaryotic cell. Okay, so option number C. The role of bacterial flagellum. So I'll just quickly tell you about this one. The cilia and flagella. I haven't told you about this. So bacteria, as you can see, they have a very small, like a kind of thread coming out of the cell membrane. Uh, so this is a see, there's a capsule, this is cell wall, and last is cell membrane. So from this capsule, this there's a small thread-like structure coming out. These small structure can be classified as fimbri or they can be classified as pilus. So this can be plural is fimbri and this is pilli. So if it is very small thread-like structure, then it is called fimbri. If it is little bit longer, but not helped in the movement, that is called pilli. Or sometime helping in the movement, not by like generating motion, then it is pilli. And the one long thread-like structure coming out from the deep inside, and it has a rotary mechanism and all, and that is involved mainly in the moving or the movement of the bacteria that is called as flagellum or as plural. If there is more than one flagellum, then it is called flagella. So three things: flagellum or flagella, which is involved in the movement, villi and fimbri, which are not involved in the movement, but they help in the bacteria to attach to a surface or stick to a surface or help in kind of moving or navigating their surrounding. These are the function of fimbri and villi, mostly attachment function. Okay, so that is the theory. Okay, so here the question is the role of bacterial flagellum. So as I just now said, which is the role of the bacterial flagellum? Does it provide structural support? Does it synthesize protein, protect cell from osmotic stress, or locomotion and chemotaxis?
Option A, sir.
Option A, correct.
E.
Yes. So facilitates locomotion and chemotaxis. Chemotaxis is something like the bacteria moves from one chemical environment to another chemical environment. Chemical environments, one suitable environment to another suitable environment. If environment B is more suitable than environment A, the bacteria will try to move to that environment B, and that is called chemotaxis. And this is achieved by motion. So flagella. is responsible for? Option A. Okay. So the other question, the component of bacterial cell wall that is targeted by penicillin. So penicillin is an antibiotic. What do you think that is there in bacteria that this penicillin targets and not in other cell? Is it capsule, plasma membrane, lipopolysaccharide, or peptidoglycan?
Sir, I think option D.
Option D. Okay. Okay. Others, can you think? Like, let's say you are getting a bacterial infection. What should the penicillin target that it won't damage your cell, but it will damage the bacterial cell? Yes, correct. So it is peptidoglycan because the bacterial cell has peptidoglycan. The animal cell does not have peptidoglycan. So the penicillin targets the peptidoglycan, which kills the bacteria. And this is how the peptidoglycan layer looks like. So we have, like, as you know, the cell wall of bacteria is composed of peptidoglycan. We have gram positive and gram negative bacteria that retains the gram stain is the gram positive because it has a thick layer of peptidoglycan. This you can see, this is the peptidoglycan. And in the starting, I said it is a like long polymer of NAG and acetylglucosamine and an acetylmuramic acid, long polymer of NAG-NAM chains. And here you can see how these NAG-NAM chains are organized and how they are cross-linked among each other. And this is the microstructure of a peptidoglycan layer, and that this is targeted by the penicillin. Okay. One more question. The protein are transported from the endoplasmic reticulum to the Golgi apparatus through what? I discussed this. Lysosome, vesicle, diffusion, or nuclear pore.
Basically, sir.
Vesicles, correct. Yes. So here I told you that in after the protein exits or done with the rough endoplasmic reticulum, it is transported in the form of vesicle towards the Golgi apparatus and it fuses with the cis phase of the Golgi apparatus. Okay, so vesicles. Okay, the feature of lysozyme or the lysosomes, what we discussed, that allows it to protect human from bacterial infection. So it inhibits bacterial DNA replication or disrupts the plasma membrane, or it cleaves the glycosidic bond in the peptidoglycan or prevent the bacterial chemotaxis. So which one do you think? Lysozyme has what that it function that this can do this particular choice?
Sir, option C.
Option C, correct. Others also try to put in the chat box, try to answer. It's fine if it is wrong. So it is option C. So as I said, as we discussed in the lysozyme, sorry, this lysosome, we have a lot of enzymes, or that is hydrolytic enzymes like lipases, proteases, carbohydrate, and these hydrolytic enzymes can digest lipid, protein, carbohydrate. Since the polysaccharide membrane is made up of lipid like magnesium chains. This can this peptidoglycan layer is made up of your magnesium chains or mostly glycosidic bonds are there. So if this has so much of enzyme, it can easily digest the peptidoglycan in the case of bacterial infection. So that uh, justifies the answer. Okay. So the plant cell have a larger vacuole compared to the animal cell because to assist in cellular division, to increase the rate of photosynthesis, to store toxic substance and maintain turgor pressure, or to provide energy for cell motility. Which one?
Option C.
Option C. Okay, correct. So, as I said, it is stores toxic substance. And what is this turgor pressure? Just to revise this concept. So, water and all sap and toxic material moves into the vacuole. And as I said, the vacuole can be really big in case of plant cell. And if there is some pressure from the outside, there should be some pressure from the inside of the cell to maintain uh like uh, what do you call that, equilibrium or stability, so that the cell I do not shrink or burst. So that is where the pressure from the inside, which is exerted by the vacuole, is called turgor pressure. So that is the role of vacuoles. Okay. Another question, the function of peroxisomes in eukaryotic cell. Specialized lysosomes, they are peroxisomes. They break down fatty acid detoxification of hydrogen peroxide, or protein synthesis, or energy storage, or photosynthesis. What is the function of peroxisomes?
Sir, option A.
Option A. So peroxisome are organelle that are found in eukaryotic cell. Remember, these are only found in eukaryotic cell and are responsible for a variety of cellular function, including lipid metabolism and reactive oxygen species, ROS, like raw species detoxification. In case of like hydrogen peroxide. So these are the peroxisomes. Okay, now I'll quickly tell you about the cell cycle and then we'll be done with today's session. So, cell cycle is the sequence of event by which a cell duplicates its genome, synthesizes the other constituent of the cell, and eventually divide into two daughter cell, is termed as the cell cycle. Here you can see a cell cycle going on. So it first divides its genetic material and then the cell itself divides. So it can be further divided into uh, two parts. One is interphase, one is M phase or the mitotic phase. So interphase is the preparatory phase or the phase in which the cell prepares for the cell division, and the M phase or the mitotic phase is the phase in which the actual cell division takes place. So this interphase can be further divided into three more parts: G1 or the gap 1, S or synthesis, or G2 or the gap 2 phase. So this is the overview of the cell cycle. Interphase is where the cell prepares for division. M phase, at the mitotic phase, where the actual division takes place. So to give you an overview of how long this preparatory or the interphase can last, that in a 24-hour average duration of cell cycle in a human cell, cell division properly lasts for only about 1 hour, and the rest 23 hour is involved either or dedicated to the interface. So 23 hour the cell takes for preparing for the division, and the last 1 hour is just the division. So you can imagine how long it takes for the interface. Okay. So we have the cell cycle, and I told you about the G1, S, and G2, and the mitosis stage. So most of these things are like you have to go through them by yourself. There's nothing much to discuss here. Like the G1 is a phase is where the cell is metabolically active and continues to grow but does not replicate its DNA. Only the DNA is replicated or duplicated in the S phase or synthetic phase. That is why the name is synthesis phase or synthetic phase. So DNA synthesis or replication takes place if the initial amount of DNA is denoted as X, then it increases to 2X. And G2 or the gap 2 is the protein synthesized in this preparation. So G2 is the last step in the interface, and then the cell will start to divide. So this protein which are required for the mitosis are synthesized in this G2 phase. So after this G1, S and G2, the cell undergoes division or the real mitosis. And then we will see in mitosis, we have prophase, metaphase, anaphase, telophase, and then cytokinesis or the cell division. And in the case of meiosis, we'll have two of this. Two like meiosis one, meiosis two. So two prophase, two metaphase, two anaphase, and two telophase. And then if the cell decides not to divide further, then it can go to a quiescent stage called as G0 and stays only metabolically active without any division. If it again decide to divide, then it can again go to this G1. From there, it can go to S from G2 and then go to cell division. Okay. So I'll tell you about this mitosis and meiosis, but not like reading it out. I'll just open the paint and I'll draw the chromosome structure and I'll tell you about the mitosis and meiosis. Just a basic overview here. Meiosis, mitosis is something called as somatic cell division, or here your cell duplicates to, like let's say there's an injury or to form or wound healing. So the adjacent cell needs to look alike the cell which has been damaged. So that this is called somatic cell division or the body cell division, and this is formed by mitosis. So whatever the cell was looking like or the parent cell, you will get exactly two looking alike daughter cell. And meiosis is something called as gametogenesis or the gamete forming cell. Because in the sexually reproducing organism, we are diploid organism, and since we are diploid organism, our gamete has to be haploid. And in the sexual reproduction, there is fusion of these two haploid gamete, and this results in the production of a diploid organism. So whatever genetic content is here in mitosis, the same genetic content will be there in the two daughter cell. But whatever genetic content will be here in the case of meiosis, these all the four daughter cell will have half of the genetic material. So if we have like 2n here we will be having 2n. Uh, if here we'll be having 2n, here we'll be having n each, n. So haploid content of the genetic material is denoted as N. I hope you all know this. N is denoted as the haploid content of the genetic material. Since we are diploid organism, we say we are 2N. Okay. So I'll okay. Before I open this paint and draw this mitosis and meiosis for your simplification, I just want you to get clear with this concept that we are a diploid organism, that is 2n. We have two copies of chromosome, like chromosome 1, chromosome 2, chromosome 3, till this 22 pairs of autosome, and the 23rd one is the sex chromosome, XX or XY. So we have two copies of each chromosome, one from the father side, one from the mother side, one paternal, one maternal. And we are diploid organism. We have two copies of chromosome. And in haploid case, there's only single chromosome, so denoted as N. So I want you to first remember this, that what is diploid, what is haploid, how does it look? So each chromosome, I told you, each chromosome is a condensed form of the genetic material that is going to divide. So genetic material in case of human, it is DNA. So DNA is something which is double stranded. DNA is not single stranded. So here we have from the paternal side, we have the DNA like this. From the maternal side also, we have a DNA like this. A DNA which is condensed and there's a histone protein which bound the DNA and condensed them, and pictorially it looks like this. One from the mother side, one from the father side. Now we will see how uh, your okay, this mitosis and meiosis is happening. Okay, I'll just open this. Okay, so let's first talk about mitosis. Remember this, before the prophase, metaphase, anaphase, telophase, that is the mitotic phase, we are done with the G1S and G2 phase. So in S phase or the synthetic phase, the DNA has duplicated or the DNA has doubled. So let's say we are a diploid organism. We have the DNA in the paternal side. Of course, in the paternal side, there will be, so the DNA looks like this. It is a double, you all know DNA is a double stranded structure. It is not a single stranded structure. And let's say in the maternal side, it is like this in the red color, different color to so paternal and maternal. Now in the synthetic phase, what will happen? Synthetic phase, there will be duplication. So the black will separate like this, the black will separate like this. So one more black will form because DNA duplication in the synthetic phase. The same thing will happen for the maternal side also. Okay, now whatever we had started has now doubled, simply doubled. Next, in the case of mitosis, we need to preserve this type of uh, configuration which was present previously. So what to do? Attach the centriole here. So the centrioles will attach. Okay, centriole, maybe I'll draw a different color. Okay, here the centrioles from the centrosome, sorry, the centrosome, it will attach. From this also, it will attach. Here it will attach. I'm not like drawing very great diagram, just to understand how the division is happening and what defines mitosis and what defines meiosis. And then you will have one black coming here, one red coming here, one more black here. Okay, I'm drawing below this. One black here, one red here. Now, without any further division, let's just put boundary around them. Let's just put boundary around them, or nuclear membrane forms, and then these become two daughter cell. So now see, whatever the parent was, the two daughter cell look alike. Told you that the centrosome they are attaching and all. These are prophase, metaphase, anaphase, telophase. I'm not going to tell you about that. You can simply open any textbook and read about what is happening in prophase, what is happening in metaphase. Metaphase, there is metaphasic plate formation and all. Simple things, normal theory. I just want you to draw the diagram and know yourself that how this mitosis is allowing to produce daughter cell which is exactly equal to the parent. So if this is 2n, so if this is 2n, this is also each chromosome is 2n. So this is mitosis. Now, okay, everyone is clear with this mitosis? And just drawing simple diagram to make you understand. Yes, shall I tell you about meiosis? People, are you following?
Yes, sir.
Okay. Okay. Like you have a look here and then later you can draw this because this video will be anyway available to you later. You just follow like how this is happening. Okay. So mitosis is done. Okay. I'll erase from here. Okay. Okay. Now, in case of what is happening in the meiosis. Meiosis, what is the difference? That the prophase, metaphase, anaphase, telophase will follow twice, or the this M phase will be followed twice. But before M phase, we will have G1, S, G2. So in the S phase, your DNA genetic material will be doubled. Similarly, in the prophase, it will come, it will attach, and it will separate, and you will get these two type of things. One black, sorry, one red, red, another black. Interestingly, in mitosis, the after prophase, metaphase, anaphase, telophase, it was over, and then it formed another cell. But meiosis, one more time it will go. Prophase 2, metaphase 2, anaphase 2, telophase 2. That means your centriole will come and attach here, attach here for this case, or maybe attach here and attach here, and they will again pull in the metaphase. So ultimately, what you are getting? You are getting, say, one black. Okay, and one red. Oh, okay, I'll erase it and make it more clear. Okay, see. So let's say one set, like it is pulling from here, one black, one red. Here it is pulling one black, one red. So how it will come? One black here, one red here, one black here, one red here. For this one red here, one black here, one red here, here. Now you form the cell. Now you form the cell. Because There's nothing called as prophase 3, metaphase 3, anaphase 3. We are done with the meiosis 2. Now you form the cell. Now you see there's twice the genetic material of each. So it is called 2n. But here we are ended up having after meiosis 1, this is meiosis 1, meiosis 2, we ended up having n or haploid content in each. And they, how many there are? One, two, three, four. So this from this you can figure out that how meiosis generates haploid cell, and these are nothing but gametes, or the process is called gametogenesis. So we started from diploid content, but we ended up having haploid content. So these are single stranded. Don't think that they will form, uh, I mean, bond with each other. One is from parental side, one is from maternal side. They won't be, they will be remaining like this until they cross over or they form the bonding with the another. Like if this is sperm, so they with the egg, they will come. If sperm and egg will fuse, they will again form this type of structure. So this is the meiosis. Okay. This was a very not so great diagram in Power Paint, just to tell you the segregation of the or the separation of the chromosomes, the sister chromatids. A very nice picture and very nice like writing you can find in any textbook. But you should understand how this is happening. So meiosis in the Okay, now we are done with this. We'll go to the theory. I hope you all understood. It is not very difficult. So this is what I told you about meiosis in simple picture. I have put prophase, prometaphase, metaphase, anaphase, and telophase. Please go through this. These are very self-explanatory. I don't need to tell you about this thing. I just told you about these things, that how this division happened. Okay, and this is about the meiosis. Meiosis. See, after the duplication in prophase 1, there is something called as crossing over. This is a characteristic feature of meiosis which happens in the prophase 1. And prophase 1 is the longest phase of meiosis, because the prophase 1 of the meiosis, only this much part I'll tell you, because this is kind of important thing, that other division remains the same. This crossing over is a characteristic of a prophase, and this crossing over of the sister chromatid, non-homologous sister chromatid, is what results in the variation, crossing over and variation. So the meiosis, this prophase 1 is divided into leptotin, zygotin, pachytin, diplotin, and dikinesis. So during leptotin, the compaction of the chromosome continues. Then comes the zygotin. Zygotin is the stage where the pairing together, and this process is association is called synapsis. So in zygotin, the chromosomes start pairing up together. These are they will start to pairing up together, but no crossing over. They will just form the pair. Pairing of each other will happen in the pachytin. So pachytin, during this stage, the four chromatid of each bivalent chromosome. So one chromosome has two chromatids. So four chromatid of the bivalent chromosome, because they have paired up together in this zygote stage, become distinct and clearly appear as a tetrad. And this stage is characterized by the appearance of recombination nodule. See, recombination nodule, the site at which crossing over occurs between non-sister chromatid of the homologous chromosome. So, non-sister means this is a separate chromosome. This is a separate chromosome. But So that is they are non sister, but they are homologous because they are of the same like category. So you let's say you have chromosome 1. So chromosome 1 cannot be going and crossing over with chromosome 2. So they have to be homologous chromosomes. So the crossing over is the exchange of genetic material between two homologous chromosomes. So this crossing over is also an enzyme mediated process, and the enzyme involved is called a recombinase. So crossing over leads to a recombination of genetic material on the two chromosomes. Recombination between the homologous chromosome is completed by the end of Pachitin, and then this diplotin starts. Diplotin recognized by the dissolution of the synaptonemal complex, so where the synapses or this crossing over is going on. It is called synapses, and the complex uh, or the cementing material is called synaptonemal complex. So this dissolves in diplotin and forms from each other. The homologous chromatids separate from each other except at this synaptonemal complex. So since they are completely separate from each other, but they are still linked at this synaptonemal complex, uh, they look like a X-shaped structure and they are called chiasmata. And in the last step of the prophase one, that is dikinesis, this chiasmata, this is the only crossing of this, the sticking site gets dissolved, and you have the two separated sister chromatids. During this phase, the chromosomes are fully condensed, and the meiotic spindle is assembled to prepare the homologous chromosome for the separation. By the end of dikinesis, the nucleolus disappear and the nuclear envelope also breaks down. Dikinesis represents transition to the metaphase. Now the centrioles can come and attach and they can form the metaphasic plate and then they can do the separation. Okay. So this is the pictorial representation. See, this is the site of crossing over. This is after crossing over. One part, like they just exchange a small part from each other. These are homologous chromosome and they are non-sister chromatid and they just exchange one part by the recombination. Enzyme involved is recombinase. And whatever the diagram I did, it explains how you get single uh, like from 2N, how you will get N or haploid content in the meiosis. Okay, please download this and go through this by yourself. There's nothing much to like explain here. These are kind of self-read things. Whatever explanation part I already showed you in that Venn diagram. Okay, so yeah, we'll solve some few more questions and then we'll be done. Anyone would like to ask anything now? This mitosis and meiosis, I hope you have already studied in 11th and 12th. Okay. Okay, so answer these questions and we'll be done. So first one, meiosis is critical for sexual reproduction because it ensures genetic diversity, it prevents the fusion of gamete, it doubles chromosome number in the gamete, or it produces identical copy of parental cell.
Sir, option A.
Option A, yes. So since recombination is happening, we are getting this genetic diversity. Recombination leading to variation. Option A. Next one, the phase of mitosis during which sister chromatid align at the cell's equator. Prophase, anaphase, metaphase, or telophase, in which in the center all the chromatids align in the cell center. It is called a dash plate. So it is called metaphasic plate, in which all the sister chromatid align at the cell's center or the cell's equator, because it is now attached to the centrosome, and they are now the centrosomes are pulling the sister chromatid. So they will align in the center, forming a metaphasic plate. So that is option C, metaphase. Okay. The next one, the process that is responsible for the exchange of genetic material during meiosis. Is it cytokinesis, chromosome condensation, DNA replication, or crossing over?
Crossing over,
Cross,
sir.
crossing over, correct. So crossing over is happening in the prophase one, is is responsible for the exchange of genetic material during meiosis. Okay. This one, another question. The decision to divide the cell occur at what? G1, early S, late S, or M? Whether the cell should proceed to divide or not. This decision is taken in which phase?
G1 phase.
G1 phase, correct. So I'll show you again here. In this phase, Z1, either if the cell is okay to divide, then it will proceed from G1 S to G2, or if it is not okay to divide, it just want to stay as it is, it will go to a quiescent phase, which is called as G0. So that will have that decision is taken in the G1 phase. Okay. The role does the spindle fiber play in the cell division? What the spindle fiber does? It synthesizes ATP for mitosis, or it separates chromosome during anaphase, or it creates the nuclear envelope, or it regulates the cell cycle checkpoint.
Sir, option B.
Option B. Okay, it separates chromosome during the anaphase because it goes and attaches to the cell. Okay, okay. Ignore this. Let's say these are the two sister chromatids. Here your spindle fiber will attach, and the site of attachment is called, what is the site of attachment is called? Come on, people, try to remember what is the site of attachment where the spindle fiber goes and attach to the chromatids.
Centromere.
Centromere is the entire location. This is the central part. So centromere has something to which it attaches. Starts with K.
Kinetochore.
Kinetochore, correct. So centromere is the entire central part. So centromere has kinetochores. Kinetochore is where this spindle fiber goes and attach. Okay, that's all. So we are done for today. I hope you people have understood this.